Buratowski as a speaker today. And Steve got his undergraduate degree the summa cum laude from Princeton University in 1984. And then he did his work with the Nobel laureate Dr. Phillip A in MIT. He got his PhD degree in 1990. And then after spending 4 years as a Whitehead Institute Fellow, and he joined the Harvard Medical School as associate professor in 1994, I think. Right? And then he became a full professor of Harvard Medical School in 2002. And then during his tenure at Harvard Medical School, he's been studying the mechanism of transcription and gene regulation by RNA polymerase II, and this also many transcription activated regulators. And also he's been studying how transcription regulate and coordinate and couple it to chromatin regulation and RNA processing. Recently, he's been adopting a single-molecule technology to more dissect the transcription initiation process. And then I guess he's going to talk about that today, right? And then so far he's been publishing more than 130 original papers in high-profile journals, and also he got many awards, including Pew Scholar Award and Nuclear Information Scholar Award. And then actually, Steve was my postdoctoral mentor when I was a postdoc in his laboratory, and then he was a great mentor, but also he provided me a lot of good environment. And I can really enjoy doing science. So I really appreciate his support. With that, please take your lead.
Oh, thanks so much. Thanks, Minkyu. Yeah, it's very nice to be here. Thanks for coming out. And I guess there are some folks online. Welcome. Yeah, Minkyu did some really pioneering work in my lab. If you didn't know, he was the person who essentially discovered the torpedo mechanism of transcription termination and some work on CTD phosphorylation. So it's great to see him progressing and being so successful. So I wanted to tell you today about, I changed my title, I think, from what I may have given you. I want to talk about models and how we build models and how models need to be updated over time. And just as an analogy, I've been starting my talk with this figure, this photograph of the statue of John Harvard. So if you come to Harvard College in Cambridge, you'll inevitably see this statue. And we call it the Statue of Three Lies. And the reason we call it the Statue of Three Lies is it says that John Harvard is the founder. John Harvard actually did not found it. He gave a big donation. So just like this building, if you give enough money, you can get your name on things. That was true even in the 1600s. They actually list the date of the donation at 1638, but they actually claim it as the founding date. It was actually founded 2 years before the donation. But the worst of the three lies is they made this statue around the 150th anniversary of the university. They had no pictures of what John Harvard looked like. And so they actually just picked some handsome looking guy to be the model. It's not really even John Harvard. So, but it's a nice illustration of how, we can come up with models and they can be very useful, but at best they're going to be approximations of the truth. And this is certainly true in molecular biology. So, as an example of how models evolve, I'll tell you a little bit later about how RNA polymerase II initiation complexes assemble. And back when I was a graduate student, we did some work suggesting this idea of a hierarchical assembly. We've gone from these kind of balloon diagrams to atomic level resolution. The structures get better and better, but even the best structure that have come out, there's still approximations because there's often big pieces missing, flexible regions that we know are important. When you see these fixed structures, you don't get any sense of dynamics. And of course, movement is really critical for biological function. So what I want to do today is sort of revisit some models that appear in textbooks and we teach it to our students, but they really need to be revised. So based on some data coming out of my lab and others, we started to rethink some of these things that we take as gospel truth. So just as a way of background, if you're not aware, RNA polymerase II, which makes messenger RNA most small non-coding RNAs, it looks very much like Pol I and Pol III, but it has this extra domain called the C-terminal domain. The sequence tells you right off the bat, this is a site of phosphorylation. It's all phosphoacceptor residues except for two proline pairs. when the polymerase gets to the promoter, it comes in the unphosphorylated form. And then it was known very early on that when the polymerase is transcribing, it's actually highly phosphorylated. And then work in my lab around 2000, 2001 made the discovery, which I shouldn't have been surprising, but it was a big surprise at the time, that the different phosphorylation sites are utilized differentially. And so in particular, serine 5 phosphorylation marks polymerases near the promoter, and serine 2 phosphorylation marks them further downstream. And because we had established this using chip experiments, we kind of defined the model in terms of the polymerase position along the gene. And that made a lot of sense because we had discovered this pathway by looking for how capping enzyme interacts with polymerase. And it turns out capping enzyme very specifically recognizes the serine 5 phosphorylated form. Later on, work from Minkyu and others in my lab and other labs found that the serine 2 phosphorylation was used for recruiting some of the 3' processing enzyme. So this idea of the CTD phosphorylation as a measure of distance made a lot of sense. We wanted to create an in vitro system to study this process, and we use a system that's been around for a long time called a mobilized template assay. Essentially, we take a little promoter DNA. It's got some Gal4 binding sites, the CYC1 core promoter. We use a G-less cassette for stalling it, put it onto magnetic beads and incubate it with yeast nuclear extract, some Gal4 VP16 activator. We can assemble PICs in the absence of NTPs, and we've done some mass spec analysis published quite a while ago now about the components within the PIC. But the idea was that we would use this G-less cassette, and we would make G-less cassettes of different sizes and then give the polymerase 3 nucleotides so that the polymerase would stall at the end. And we would do a short cassette for early complexes, a little bit longer one for middle, and then a longer G-less cassette for downstream. But when we did the mass spec analysis, we always got what looked like a late elongation complex, something that would have been down near the 3' end. And we kind of scratched our heads for a little while and then had one of those light bulb moments. It's like, well, wait a minute, what if it has nothing to do with the distance traveled, but what if it's actually a timing mechanism? So Eugene Ju, who did these experiments, redid the experiment where we took really quick time points before we were incubating for 30 minutes. But if you do these quick time points, you just get a phenomenal result. This is still one of my favorite results ever from my lab. When at the 0 time point, when you assemble the initiation complex, the polymerase is there. And the minute you add the nucleotide triphosphates, you get really strong serine 5 phosphorylation. Within a few minutes, that's fading away. And you come up and you get the serine 2 phosphorylation. So it matches perfectly what we wanted to see from the chips. But this polymerase is only going 200 or 300 nucleotides. And if you look at the transcript, by a minute or two, it's at the end. So that CTD cycle is going on, even though the polymerase is not physically moving forward anymore. So it's really measuring the time from initiation, not the distance. And that changes the way that you think about some of these models. And we can see that, that very nice correspondence. We had been defining the experiments in terms of the chip distance, but the distance along the gene really is just a proxy for time. And we can see that with the in vitro system. We know this is true in vivo because we can do some ChIP-seq experiments. This is some work done by Luis Suarez. If we look at the serine 2 phosphorylation in different yeast strains where we either have wild type polymerase or slow polymerase or fast polymerase, these are point mutations in the active site that affect the elongation rate. We can see that the slow polymerase shows CTD serine 2 phosphorylation much earlier, and the fast polymerase takes a little bit longer and peaks further downstream. So that, that model about CTD measuring distance, we've updated that now, and that it really has to do with time. And that turns out to be critical if you think about polymerase pausing, right? So there's this idea that in many metazoan cells, the polymerase goes for maybe 50, 60 nucleotides, and this sits there paused for quite long times waiting for a signal. If that's true, then you've got to think about what it is that stops that CTD cycle from moving forward. Another explanation, which I think is really the proper one, it's become more and more clear that these so-called pause polymerases are actually terminating, and they never get released into elongation. There's work from even Karen Edelman, one of the big proponents of the pause model, and other groups showing that those polymerases at the 5' end do turn over, they cycle, spit out these short RNAs, and never actually get released into elongation. So that may be a part of what we've been calling pausing all these years. The second model that I want to tell you about has to do with histone modifications. And it's been known again for a long time that if you look at active genes and you look at some of the histone modifications, there's this very characteristic pattern of K4 methylation near the promoter, K36 methylation downstream. And work that we did with Nevin Krogan and many other groups showed that that's mediated by direct interactions between the two methyltransferases, set1 and set2, binding respectively to the serine 5 or the serine 2 phosphorylated forms of the CTD. And one of the things that we found curious and others found curious was the fact that even though there's a single methyltransferase in yeast, set1, you get differential methylation levels as you move from the 5' to 3' end, tri, di, mono. And there are a few models that are out there. One of them is that the polymerase, that the set1 complex dissociates as the polymerase moves from 5' to 3' end. And that can work if you can imagine being, mostly at the promoter putting on 3 methyls. If it starts dropping off here to dimethyl, you can explain that transition. But of course, if you're dropping off and only making dimethyl, then the question is, how does the methyltransferase get further downstream to do monomethyl? So that model doesn't quite explain things. And then there was this other model that the enzyme was actually somehow allosterically regulated, that somehow it would know where it is along the gene and be more catalytically active at the 5' end versus the middle versus the 3' end. So we thought about these models, and this again was work by Luis Suarez when he was in my lab. Luis did some ChIP-seq experiments, and one thing immediately jumped out that kind of also ruled out a simple allosteric model, and that's that there are genes that don't have the canonical pattern. On most genes, you see tri, di, mono spread out just like you expect from the metagene analysis. But down here, you'll see there are hundreds of genes that have mostly dimethyl at the promoter, very little, if any, trimethyl, and even a few that have monomethyl, but no di or tri at the promoter. So how do you do that? Well, one way you could do that is by demethylating. We've knocked out the demethylases. That's not an explanation for what makes these genes different. So another argument that there's positional information comes from this experiment, similar to what I showed you before. We can do the ChIP-seq for the methylations using either fast or slow polymerases. And we can really see the difference here if we look at the dimethyl. So the dimethyl with the slow polymerase is here a few hundred nucleotides downstream, with the fast polymerase much further downstream. I'm not showing you the wild type, but that's going to be right in between. And even for the K4 trimethyl, we can see the fast polymerase draws it down much further. So the fact that the methylation position is sensitive to the speed of the polymerase, again, says it can't be purely positional information determining what's going on. And so, once we started thinking about dynamics, something occurred to us. All the models that had been proposed really made this assumption that a single passage of the polymerase would set up the tri, mono, and di. And we thought, well, what's the evidence for that? There's no reason that has to be true. We know that nucleosomes get recycled as the polymerase passes through the gene. There's machinery for disassembling the nucleosome downstream, holding on to that same set of histones, and then replacing it behind the elongating polymerase. And so you could imagine that methylation is sensitive to multiple passes. Maybe the first time the polymerase goes through, you get to mono, and it takes the second polymerase coming through with a new set1 methyltransferase to go from mono to di. So we started to think about how we might test that. And one of the predictions of that kind of multi-pass model is that the methylation gradient should be sensitive to the frequency of transcription. By that, I mean how many times the promoter fires off per cell cycle or per unit time. So Luis took his data and we just sorted the genes by their expression level. And we could do either RNA levels, we could do POL2 ChIP, they all give the same result. And you can see here, particularly with the K4 dimethyl, as you go from the most lowly transcribed to the most frequently transcribed genes, that dimethyl peak shifts downstream. And you can also see that the K4 trimethyl spreads out a bit. And that's a nice explanation for how you get these sorts of things. So we've now come in and we've revised the model the way we're thinking about it. It is true that the K4 methyltransferase does drop off as you go from 5' to 3' end. If you ChIP set1, there's no question that's happening. That makes sense because it's targeted by the serine 5 phosphorylation. But now with this additional complexity of considering transcription frequency, now you can imagine that the first pass, maybe you get mostly mono, maybe a little di, second pass a little bit further up, and most genes where you've probably got multiple rounds of initiation give you the canonical pattern. One last experiment, I won't show you the data. We actually took the methyltransferase and we fused it to one of the polymerase subunits, dragging it along with the elongation complex. In that case, you see the dimethyl and the trimethyl go all the way downstream, again, ruling out positional information. Just in case you're thinking, well, yeast only has a single methyltransferase, what about metazoan cells where we've got SET1, but also MLL1, 2, 3, et cetera? We found a few papers in the literature that were looking at these unusual metazoan genes, mammalian genes, where the K4 trimethyl spread much further downstream than expected. And they didn't quite think of it the same way we did, but you can see they're sort of moving toward that idea. The genes that showed this pattern tend to be the most highly expressed in the cell. This paper came close. They used this term transcriptional consistency, which I think is similar to the frequency thing that we were thinking about. So we think this is probably a generally applicable thing to a model to use for even metazoan cells. Some of you who may work on mammalian gene expression know that K4 monomethylation is often used as a marker for enhancers. And it may be true that there are specific methyltransferases that put mono there, but many enhancers also have some transcription going on. And so those transcription units tend to be very weak. We also know that that if you look at enough enhancers, you will find enhancers where there is trimethyl and dimethyl, and those tend to be more highly transcribed. So I've never thought that there's anything particularly different about enhancer RNAs. My guess is these are mostly just weak promoters. Okay. So, I want to move on to the most recent stuff that we've been doing, which is some single molecule microscopy. Again, looking at some of these basic processes that we think we understand. And I want to start out with this question: how is it that transcription activators work? We'll have activators bound at the UAS or enhancer. And if you open a textbook, it'll say, well, the activators recruit things. They'll recruit these co-activators. They're going to recruit chromatin remodelers, histone modifiers, but also parts of the basal machinery. And so the factors binding here somehow increase the binding of factors here. And there must be some sort of connection, some sort of arrow that often is drawn. But what does that mean when you say it's being recruited? All right, so you can imagine it's sitting there and somehow helping it get there faster. In that case, you'd see an increase in the association rate. It could be that these things bind independently, but they make a cooperative interaction. So you're actually slowing down the dissociation rate. How do we start to distinguish these kind of models and get to a more precise term than recruitment? Well, and again, this is going back to this idea of models and how models force you to think or tend to pigeonhole the way you think about things. If you look at some recent reviews and you look at activated transcription, they always show something like this, where there's some sort of looping between enhancer regions and core promoter. But these models are really static. In some cases, it's proposed that these interactions are quite stable, sometimes even held together by things like cohesins. I think more recent data suggests at best these sorts of things are transient. When they show the coactivators, they tend to show everything all together in one big glob. And of course, that doesn't have to be true, probably not true. It could be that all those different coactivators that get recruited by activators are coming sequentially. They could be mutually exclusive. They could be coming together, but we don't know how to tease that apart. If you're doing things like ChIP-seq, they're all cross-linking in the same place. That doesn't mean they're there at the same time. And then finally, there's been a lot of a buzz about things like condensates and eRNA sort of holding these things together in these little globules that highly concentrate the factors. All of these things are kind of current models, but we really wanted to move beyond this. We wanted to get to a point where we could actually see these things in real time. And if you think about the time scale for gene expression, these events are happening on an order of seconds, maybe minutes at most. So that means any assay where you're doing cross-linking for 20 minutes, or even equilibrium assays where you're doing DNase footprinting, where you bind for 20 minutes and then cut, or gel shifts, all of those, the time scale is way too long to capture these things in real time. The other thing that we would really like to be able to do is look at individual molecules, right? So when we do anything like a chip or gel shift or footprint, we're looking at an ensemble and we're finding the average of the events that are going on. But if you actually have different pathways or different species mixed together, what you're going to look at is the mixture of those signals. So as one of my graduate students put it, if you have two populations, 50% yellow and 50% blue, when you do an ensemble assay, you're going to say, oh, it's green, right? Because it's mixed together. So we would like to be able to do single molecule studies, but also have enough numbers where we could get some statistics. So to make this possible, we started working with Jeff Gellis, who's on the faculty at Brandeis University, and this is his right-hand man, Larry Friedman. And these guys together have been doing single molecule work, some really beautiful work on bacterial transcription, but more recently, eukaryotic replication with Steve Bell at MIT, and mRNA splicing with Melissa Moore. And so we decided to take our in vitro system and adapt it to their microscopy system. And that work was done primarily by Inwa Baek, and then more recently by Jungchul Jeon. So we had the assay in hand because I told you before we had this nice immobilized template assay where we could follow CTD phosphorylations and all that. And so we take that same DNA, put the biotin on one end, but instead of hooking it to a magnetic bead, we're going to put it onto a microscope slide, and we've got a fluorophore on it so we can map the positions of all the DNAs. So every one of these little spots is an independent DNA. And then we can use turf microscopy to essentially illuminate molecules that are close to the slide surface. Inwa spent a lot of time working out conditions where we could look at two different proteins, one in green channel, one in the red channel. These are in yeast nuclear extracts. These are not purified proteins. We're taking the whole yeast nuclear extract, which has all the components of the nucleoplasm, we like to think, in the right proportions. We could purify things, and there are groups who do that. Purifying all the proteins needed is a heck of a lot of work, and you're never quite sure if you've reconstituted things in the right way. So using this system, we can just take movies in the green and red channel and watch the spots of the transcription factors come and go. And then the individual frames can get translated to these intensity traces. And here you see a molecule of Pol2 binding, sticking around for a while and then dissociating. Here's a molecule of TF2E in the other channel binding to the polymerase, and then they fall off together. So that's a single DNA. We can take a collection of all of these individual traces and array them in what we call a rastergram. And essentially, we just create a binary where it's either bound or unbound. If it's bound, we put a little bit of color. Here's the time axis. And then we like to sort the DNAs by the time to first binding, right? So here are the earliest binders, later. And finally, up here, we're saturating. We're getting up to 80% templates bound at least once during the experiment. And you can see, we see a mixture of short and long binding events. We see very often the same template gets bound multiple times. There's a lot of information in here. If you look at the leading edge, you probably say, oh, that looks kind of like a binding curve. And in fact, we can take that cumulative binding curve and fit a numerical equation to it from which we can derive the association rate. We can also take the dwell times and actually calculate the dissociation rate. So there really is a lot of information in here. Just to convince you that this is real polymerase associating in a transcription dependent manner, this binding is done in the presence of the Gal4-VP16 activator. If we leave out Gal4-VP16, we see no binding above background. I should say one really critical control we always have to do is have a control set of spots where there's no DNA, just so we know how much the factor kind of sticks to the slide occasionally. And so this is really clear that we're getting Pol2 association in an activator dependent manner, as you'd expect. So let me first tell you about some unpublished work that Jungchul's doing, looking at transcription co-activators. So again, the idea is that you've got these enhancers, the activators bind there, and the activator is going to recruit things like switch SNF remodelers, histone acetyltransferases, and that's going to open up the chromatin template DNA. So now you expose the basal promoter for polymerase and TF2D and the general transcription factors to get in there. These activation domains on the activators also can recruit polymerase through the mediator complex, right? So that's the cofactor that brings in polymerase. And then finally, TF2D and TBP are also thought to interact. So these activation domains, even though they're often very short, unstructured, they really potentially interact with a lot of factors. And again, we'd like to know, how does a single activator interact with all of these things? Is it the same time? Is it sequentially? Is it independent? So Jungchul decided to look at the SAGA histone acetyltransferase. This is the major histone H3 acetyltransferase in Cerevisiae. So there are GCN5 complexes in mammalian cells, P300 CBP are very similar kinds of complexes. The idea is the activator brings these in to locally acetylate the promoter, and that helps to promote transcription. So John Chol noted a few things. You can see with SAGA, we see sometimes one molecule binding. Very often we'll see a second molecule binding. You can see that increase in the intensity. If he takes all these traces and again does the rasterogram, it looks something like this. Now we wanted to see what happens as we titrate from 0 to 30 nanomolar activator, again VP16 in this case. And the first surprise was if we're doing this on naked DNA templates, we get actually a fair amount of SAGA binding. About 60–70% of the templates will bind SAGA in a completely activator independent manner. So we were not expecting to see that. But as we titrate up the amount of activator, you can see the slope of the curve increases. So there's definitely a little bit of increase in the association rate. And where we really see the strongest effect is the duration of the binding. We get much, much longer binding events when we have activator present. So the Gal4 VP16 is not only helping get SAGA there in the first place, but it's really stabilizing it once it's on the promoter. So, we worried maybe this is some kind of artifact to be binding to naked DNA. But I would point out that certainly in yeast, and I think this is true in many metazoans, when you look at promoter regions, they often are in what we call nucleosome-free regions or nucleosome-depleted regions, where there's really quite a lot of naked DNA there. So that interaction with the naked DNA might not be completely artifactual. The other place, we've noted, I mean, the field has noted for years that there's also some histone acetylation in transcribed regions of genes, right, far away from the activators. And as the polymerase passes through, that's also going to transiently expose the DNA, the naked DNA. And that's another time when it might be that SAGA binding to naked DNA becomes relevant. But John Chol wanted to go to a more physiological template. And so we worked up a system where we could wrap the DNA in chromatin. So we make some recombinant histones in E. coli, form them into octamers. We use a chromatin remodeler, histone chaperones to assemble them into what we think is a reasonable approximation of what chromatin might look like in vivo. And what's really nice about this single molecule system, we can actually look at two DNAs side by side, both naked and chromatin in this case. And the way we do that is we'll flow in the first template, map the positions, and then flow in the second template, in this case, the chromatin, and map the new positions. So we know which DNAs are naked and which DNAs are chromatin. And so now the experiment John Chol did that I'm going to show you is looking at two colors. We're going to look at SAGA recruitment in green, but we're also going to look at the activator binding in red. I'm not going to show you the full trace for the red because what we really want to see is the time of first binding. So I'm just going to show you the first few frames of the activator binding, and then on the same DNAs what the SAGA binding looks like. So here's the naked template. And here you can see this little line of dots in red. That's the first binding of activators to each of those templates. And just like before, even when the activator is not present, we see some SAGA binding. After after activator binds, we see a little bit more binding and longer duration binding. So that fits what I showed you before. But now on the same slide, side by side, if we look at those chromatin templates, we see this beautiful activator dependence for the SAGA binding, right? So clearly this SAGA binding is responding to the presence of the activator. And again, I'm not showing you the full activator binding traces, but when there's SAGA present, there's almost always activator binding as well. So this is great. I mean, this looks really like what we would expect to see based on the in vivo analysis. And we're excited to push this assay forward. We did worry about one potential artifact. It could be the case that some of our chromatin templates are getting dechromatinized, that the nucleosomes are falling off or getting remodeled. And when we see activator binding, that's also because the DNA is naked and SAGA also can bind to the naked DNA. So to rule out that this was something going on with the chromatin, John Chol did this experiment where we compared the Gal4 VP16 activator to just the Gal4 binding domain alone. And I changed the colors, I apologize for that. But here's the binding curves for the Gal4 either with or without the activation domain, basically unaffected. But when we look at the SAGA recruitment, we can see that without the activation domain, we lose that SAGA, that strong SAGA recruitment. So that tells us this is really dependent on an activation domain. And I won't show you the experiments, but now we've swapped in different activation domains. VP16, it's a viral activator. Yeast would never see it, but we can put in different yeast activation domains. And interestingly, they have slightly different behaviors. They recruit basically in the same way, but the time that the association rate increase, the dwell time increases, those all are a little bit different with different activation domains. So of course, now we're going to start looking at some of the other co-activators, and this is just some really preliminary data. Here's SAGA, again, activator in the presence or absence of activator. Here's switch SNF, one of those chromatin remodelers, and you can see it's quite a different response. Again, switch SNF has some binding to the DNA alone. In the presence of activator, we definitely see an increase in the on rate, but actually the off rate's not really that different. So quite a different response. We did mediator. Mediator kind of gave us a little bit of a shock because at the low activator concentration, we saw basically no difference at all. But if we bumped up to 100 nanomolar activator, now we got this really strong mediator recruitment. So that's really exciting. If you've followed the literature as long as I have, and maybe some of you remember terms like synergy or combinatorial activation, this idea that on many genes you use 2 or more activators, and the combination of them gives you much stronger activator than just the sum of the individual ones. And so, so we're excited to follow up on this and start thinking about things like cooperativity in the activation mechanism. So, so just summarizing this part, again, this is looking at the co-activators. We've got some new surprises, SAGA binding to naked DNA, differential responses of co-activators to the same activator. We now can say when we talk about recruitment, we can break that into components of increased association or decreased dissociation. And really this chromatin system is going to be very valuable. The chromatin that we've used so far is just recombinant. There are no histone modifications. So of course, moving forward, what we want to do is start thinking about how is this process affected by acetylating the nucleosomes? What's affected by ATP or NTPs, et cetera. And so we've got quite a lot of experiments lined up moving forward to try to tease it out some more. So let me move to the last part of the talk in the last 15 minutes or so and tell you about work of Inwa. And Inwa really was the pioneer in getting transcription to work in the Cosmos system. She's just a fantastic scientist. She got interested in PIC assembly, and remember going back, I showed you this model. So this is actually from my PhD work. And at the time, what we were doing were gel shift and footprint experiments, and we didn't have any of these factors really purified, and certainly none of them were cloned, but we had fractions where we had separated them. And we had a system on adenovirus major late promoter where we could label the DNA with some radioactivity and then do the gel shift and footprinting. And as we added back the different fractions, we could come up with this kind of sequential assembly. We would see the secretion of factors in a very defined order. But we knew even then, and when I wrote this, I was kind of careful. We said, okay, well, this could be a model for sequential assembly of factors on the promoter, but in vivo, it's certainly possible that some of these factors were pre-associated and they might come in as partially assembled complexes. And that turned out to be a model that some people liked a lot. So again, when we did these experiments, we were doing these sort of partially purified factors. There was no activator, there were no co-activators present. We were kind of reducing the system to its bare components. But one thing that didn't work well with this system was transcription activation. So people like Roger Kornberg, Rick Young, the Reinberg group started realizing there was stuff missing. And in particular, when you started isolating some of these polymerase complexes, you could find that they were in fact associated with a bunch of other factors. The one that really came out of this kind of work was called mediator. And again, mediator, if you are not familiar, is essentially the direct contact point of the transcription activation domains on one side and the polymerase on the other. It literally mediates the activator polymerase communication. But this holoenzyme kind of went further. So these experiments were generally based on co-IPs, some sizing columns and things like that. And some of the papers were assigning the name holoenzyme to combinations not only of polymerase and mediator, but many of the basal transcription factors, or in this case, the Reinberg paper, they were even finding things like nucleotide excision repair and DNA repair factors potentially associated. Some of this almost certainly turned out to be chromatin fragments and things. I mean, this sort of strong holoenzyme model I think has not held up over time, but certainly the polymerase and mediator interaction has, and possibly some of these other basal transcription factors. So this is where we go back to the single molecule, because now we can look at this directly. We can label two different factors and ask, are they coming together or separately or sequentially using Cosmos, right? And so again, the experiments I'll show you generally will have polymerase in green and one of the other general transcription factors in red. So the first thing that popped out when we did these experiments was something we were really not anticipating. We were taking a DNA fragment that had a single promoter, just the core promoter from CYC1 and those 5 Gal4 binding sites. But when we looked at the intensity traces, it was really clear we were seeing lots of DNAs binding multiple polymerases, 2, 3, sometimes even 4 polymerases on the same DNA. So how do you get multiple polymerases? Well, of course, a trivial explanation would be there's some kind of non-specific binding, but we knew that couldn't be true because all of this binding was dependent on having the activator present. Of course, we started considering some of these condensate clustering models. Maybe the polymerases were somehow associating in some kind of little liquid droplet. But the more we thought about it, and with a little bit of experimentation, we came up with what I think is a much simpler model. And that's that when we look at the polymerases bound on the template, or what I should say is that co-localized with the template, there's another way to get polymerases there. There can be polymerases that are not directly binding to the DNA, but in fact are tethered there via the activator and the mediator. And we have a lot of circumstantial evidence that backs that up. We've imaged mediator now. Mediator behaves exactly the same as polymerase, and we see a very high correlation of their presence. Basically, polymerase seems to only bind when mediator is bringing it there. And again, various other things that are in the paper you can take a look at. But the really strongest evidence is this experiment that Inma did. So we said, well, if polymerase is going to the UAS through the activator, we should be able to cut off the core promoter completely, get rid of the TATA box and see what happens. So once again, we do this 2 DNA experiment where we bind first one of the DNAs, in this case with the promoter, and then the second DNA that has only the UAS sequence. And when we look at the binding curves, remarkably, the binding of polymerase is almost completely unaffected. The initial binding of polymerase looks almost identical whether or not you have the core promoter, right? So that's a really different model than what I was telling you before. Again, we can put some numbers on this and derive the association constants, the association rate constants. And if the polymerase really could go to both locations, we would have expected the binding to this fragment with the UAS and promoter to be the sum of these 2 different rate constants. So the fact that this sum is basically the same as the UAS alone tells us that the polymerase binding is much, much faster to the activators at the UAS. And remember, all of this binding is activator dependent. And once you start to think about this model where the polymerase is first going to the enhancer, then of course you think about models where it has to transfer from there onto the core promoter. So really a different way of thinking. Now, one thing you might have noticed if you look at the rastograms is there is one difference. You can see that with the UAS alone, most of the binding events are short, whereas when the core promoter is present, we see many more of these long events. And we can graph this out essentially on what is essentially a dissociation curve. This is a logarithmic scale. So if you see a straight line like this on the UAS alone, that tells you that there's pretty much a single population of activators that are behaving and dissociating with some characteristic frequency. It's on the order of maybe 10, 20 seconds. That's the majority, but down here there's maybe 10, 20% of the events where we're seeing this long-lived population. And if I didn't mention it before, one really key point about this experiment is we're doing it in the absence of nucleotide triphosphates. So there's no initiation going on. Once we assemble the initiation complex, it should be able to sit there relatively stably. And we can see that in the trace here. So we've got these 2 modes of polymerase binding, one indirectly to the DNA through the activator, and I'm not showing you, but mediator would be here as well. And then the longer duration complex that we believe is the initiation complex. So we wanted to go on and convince ourselves that these long events really were the initiation complex. So Inma started doing these 2-color experiments where she would image both the polymerase and one of the basal transcription factors. Here's TF2F. TF2F and polymerase were known for a long time to be able to bind in the absence of DNA. If you pull down pol 2, there's some F associated and vice versa. And the traces just look beautiful. Mostly we see this where they come in pre-associated as a complex. So it does seem like Pol2 and F probably sort of form some kind of pre-complex that comes in as a unit. But there are also many events like this where we'll see the polymerase bind first and then F come in later. You'll notice that TF2F, just like Pol2, we sometimes see these multiple binders. So here are 2 molecules of Pol2 of TF2F binding. We only see multiple 2Fs if there are multiple polymerases present. So that's interesting as well. And again, raises the possibility that F might be going to the UAS, not directly to the core promoter. And so Inwa did that experiment where we compared the 2 DNAs with and without the core promoter. And sure enough, F also seems to first go to the UAS. So she's pushed that to look at the other basal transcription factors. TF2E, another factor that interacts directly with the polymerase, also seems to go primarily to the UAS before the promoter. And we started to get worried at this point, maybe there's some Tata box in the UAS we didn't see, or there's some kind of artifact. So we were very reassured when we went to the final component of the PIC. So TF2H is acknowledged as the final piece of the puzzle. And TF2H shows a very different behavior. It really only binds above background when that core promoter is present. You can see that otherwise it's basically at background levels. So the TF2H binding turns out to be interesting because this we can use as a marker for the final assembly of the PIC. And if you look at the traces with Pol2, we get nice results like this, where we'll see these intervals when Pol2 binds, and then it takes a while, usually on the order of 10 or 20 seconds before TF2H can come in. And again, you can even see that in the bulk graphing, where there's this sort of lag time, and then the specific binding comes in. So we interpret that as that time interval as the time it takes from polymerase to go from the UAS to the core promoter, because once it's at the core promoter is when H can come in. So we can use this as a little bit of a proxy for how long it takes to transfer the polymerase from the enhancer to the core promoter. So, so going back to a model, if we want to do a balloon diagram, now we're not thinking about this sort of sequential 1, 2, 3, 4, adding one factor at a time, but we're thinking about something much more branched where polymerase mostly pre-bound with F is coming in at the enhancer, although again, we sometimes see polymerase coming before F. We never see F coming before polymerase, so that's gratifying. And then presumably there's some kind of transfer from there to the core promoter. And this is something we're thinking about. How do we measure this in a more rigorous way? And that's going to be interesting to think about. TF2E is another factor that can bind at the enhancer. E is interesting and different than F in that it comes on and off quite a bit. So we can, in the later times of the traces, see some E binding later to what we presume is the partial PIC assembled here. What's nice about this is when you think about E and F are basal transcription factors in the Pol2 system. But if you look at Pol1 and Pol3, which again are very, very similar to Pol2, they don't have separate factors for E and F. But what they do have are some additional polymerase subunits that look a lot like E and F. And so in the Pol2 system, these have probably split off to be separate factors, whereas in Pol1 and 3, they may actually be intrinsic subunits. And that fits with this idea that that's the unit that can transfer to the core promoter. And then finally, H can come in about 10 or 20 seconds later to complete the PIC. So, so now I just want to do some speculation. So once we start thinking about this kind of non-linear model where some of the components are coming through the enhancer or the UAS, this I think has some interesting applications to some other observations that are out there in the field. So you may be familiar with this idea of transcripting bursting. So people who do in vivo imaging of gene expression notice something very interesting. If you look at a promoter that produces 10 transcripts per hour, it's not like it's popping off a transcript every 6 minutes. What you see is that it'll fire off a few transcripts rapidly, and then there'll be a long break, and then it'll fire off again, a kind of a burst. And so there are different mechanisms for possibly explaining bursting. We don't know the mechanism, but there's several models, and they're not even mutually exclusive. But one of them, I think, can come out of this idea. If you have a set of polymerases sort of lined up at the enhancer ready to go, then when something happens at the core promoter, maybe TBP binding or nucleosome remodeling that exposes the DNA, now you have a window. And if these polymerases are lined up ready to go, you can easily imagine how that might lead to a bursting kind of behavior. So I kind of like that idea. The other thing I think that it's really worth considering is what this means about the observation in microscopy, where you see clusters of polymerase or clusters of mediator together, right? So again, you've probably seen some really high profile papers talking about polymerase forming condensates or liquid phase separation droplets. And I'm not saying that can't be the case, but what I am saying is that just because you see a cluster polymerase, it doesn't have to be that way. So in our simple system with 5 Gal4 binding sites, we can see 2 to 4 polymerases all bound at the same time, right? So if we were looking in the microscope in the cell, you would see a cluster of 2 to 4. If you think about enhancers in metazoan cells, which have many more activators, 20, 30 sometimes, you again can imagine that you would see a cluster in the microscope. And if you dig into the papers and you sort of look at the numbers from papers from Darjak and Sissi, Pertsinides, the numbers are, not the same as you'd expect from in vitro experiments where people do condensate formation in vitro. Those experiments, you're often taking high concentration of purified proteins, sometimes at low salt, and you get these droplets. I mean, we're talking hundreds of thousands of molecules generally in those sorts of droplets. And in the in vivo observations, often these clusters are small. They're really fairly short-lived, on the order of 5-10 seconds. Most of them. There is a subset, a very small subset, that have much longer lives and are much larger. But the vast majority of them are small, transient. And when you look at the intensities, probably on the order of maybe 10 to 20 or so molecules per cluster. And so I just want to throw that out there, that just because you see a collection of these molecules together in vivo, you don't have to invoke any kind of condensate mechanism. There's a very simple explanation based on DNA binding that provides an alternative model. Of course, what we want to do now is move on from initiation to elongation. We actually did publish one paper in PNAS a few years ago where we co-image RNA polymerase II and SPT5. This was done by Inwa and Grace Rosen, a graduate student in Jeff Gillis's lab. And the reason these experiments are really critical, I just want to point out here, is because a lot of our models about elongation are based on chip experiments. And so you could imagine an elongation factor that binds to the early elongation complex and then just sort of stays associated throughout elongation. But you could also imagine a mechanism where the factor is exchanging on and off rapidly as the polymerase moves from the 5' to 3' end. If you're just doing chip experiments, these are going to give you exactly the same pattern. You're going to see polymerase that's going to be transcription dependent, and it's going to go from the 5' to 3' end. It's even more complicated when you think about multiple elongation factors. So we know about a half dozen or more elongation factors that show the same chip pattern from 5' to 3' end. They may all be there at the same time. There are some structures now where we can see these things coexisting on the polymerase. Or they could be coming on and off, maybe one binds and recruits the other. They could be sequential. They could be mutually exclusive. There could be 2 flavors of elongation complexes. That is never going to come out of genomics and cross-linking. You really need to do this kind of single molecule work to pick that apart. So just the last data slide I'll show you, this is polymerase and SPT5. We always see a single SPT5 bind per template. We never see more than one. SPT5 binding is completely dependent on the presence of RNA polymerase. SPT5 binding is completely dependent on the presence of nucleotide triphosphates. So we really are quite convinced these are elongation complexes. And essentially, the behavior we see is pretty much a single SPT5 binding event per polymerase binding. Once in a while, on long-lived polymerases, we might see a little bit of exchange, but for sure, it's not coming on and off rapidly. Just in thinking about timescales, the lag time from the polymerase binding to SPT5 is usually on the order of about a minute or 2 when we look at the cumulative curve. So now we can start to piece in our mind in the model a rough timescale, right? So we've got polymerase coming in first, looks like mostly at the enhancer. Roughly 10 or 20 seconds, we see TF2H come in, right? And so that's the signal. Again, we're kind of thinking that's probably roughly the time it takes from polymerase to get off the activator onto the core promoter. And then for SPT5 to show up is maybe another minute or so. And so again, we can start to assign some timescales to these critical events going from activation to initiation to elongation. So we're really excited. I'm really excited about this approach. I mean, I've been working on this problem again since I was a graduate student decades ago, and to watch these things in real time. I mean, actually, when you watch it on the microscope, you just see these little blinking lights. But when you look at the data, it's so far from where we were back when we didn't have any of these things purified and cloned. We have tons of ideas of things we'd like to do. You know, we want to look at other factors as well. We want to start thinking about chromatin modifications and interactions with acetylated nucleosomes. We would really like to start looking at CTD, phosphorylation events, and the factors that bind to them. I have a new person in the lab, James Portman. We're going to try to do some experiments where instead of just tethering the DNA on one end and saying is a factor bound or unbound, could we actually make a really long transcription template, stretch it out so that we could see the promoter at one end and maybe watch the polymerases and other things move from one end to the other? That would be really awesome. So that's one of the things we're hoping to do. And then, of course, the technology we would love to improve on as well. You know, if we could get 3 colors of proteins in there where we could watch 3 at a time, that would really improve our throughput. We can easily combine these kinds of things with FRET studies where we can measure very close distances, et cetera. So there's a lot to do. I don't know if there's any grad students in the audience or watching online, but we would love to have some really motivated people. These are challenging experiments, but they're really powerful, and we're looking for people to bring us the things they're excited about to see if we could work all that in. So let me just leave with this final little reminder, and maybe it's a little bit pedantic, but I think it's really important now to start incorporating dynamics into how we think about transcription and other events as well, because we don't want to get locked into these kind of statue models. It's been useful, and it is useful for many things, but we have to progress beyond that, where John Harbert here is up and moving around and donating money or whatever it is he might want to do. So, so this is the current group, and there's Jungchul. Some of these folks are doing single molecule stuff. Some are doing completely different things I don't have time to tell you about, but I'll thank them. I'm going to thank Jeff and Larry again, our collaborators. Thank you for showing up, and be happy to answer some questions. Yeah, so there's a bunch of data in the literature, and especially in the last few years. So in the yeast system where we work, we've known for a long time about what's called the early termination pathway. This is again some work that Minkyu and other people in my lab did. So in yeast, there's a termination pathway used for small non-coding RNAs, SNA RNAs, SNO RNAs that don't get polyadenylated. And in that case, it happens very shortly after the polymerase leaves. Most of these non-coding RNAs are maybe 100, 200 nucleotides. And so in that system, every time an mRNA is getting made, the polymerase has to pass through this early termination window, and there's mechanisms to do that. In metazoans, that hasn't been so clearly the case until recently. So there are a couple of newer complexes that have been discovered. There's something called integrator. So integrator, just like our yeast system, was originally characterized as the termination pathway for short non-coding RNAs that don't get polyadenylated, but work from Michelle Pagano, Karen Edelman, various other people have now shown, even at many mRNA promoters, most mRNA promoters, the polymerase has to pass through this early termination window when integrator could be terminating it. And if you knock down some of these integrator subunits, now you see, instead of that big peak of pause polymerases, a lot of the polymerases go downstream. So for sure, some of the cross-linking in RNA that people have seen at the 5' end is not because the polymerase is sitting there, it's because it's cycling through this early termination pathway, and only a fraction of them make it through to further elongation. Mm-hmm. So I suspect what happens is, and so you might be thinking things like PTFB, CDK9. I mean, essentially, those probably make that polymerase resistant to the early termination pathway, right? So if you do a CDK9 inhibition, you see a lot of polymerase piling up at the promoter and never extending downstream. There's a factor called NELF, negative elongation factor, that was supposed to be the pausing factor. But ironically, if you knock down NELF, you don't see polymerases going downstream, right? So that tells you that that model of NELF holding it there can't be right. But that doesn't mean the CDK9 phosphorylation isn't, instead of the polymerase going, waiting for CDK9 to show up, it may be the case that if CDK9 gets to the polymerase within that early window, now it becomes resistant and passes through. That's my favorite way of thinking about it. Yes.
Beautiful work.
Thank you.
I'm curious about your experiments on chromatin.
Yeah.
A preliminary question would be how is GAL4 overcoming the barrier to repression
Yep.
posed by these nucleosomes?
Mhm.
Is there switch SNP activity in your nuclear extract that is sliding and evicting nucleosomes that enable activator binding, and then you have some subsequent series of events?
Yeah. So when we do this just with GAL4 activator alone, we see binding. So I think, some people might say GAL4 is behaving as a pioneer factor where it can get into DNA even if it's wrapped in nucleosome. We don't know the exact position of our nucleosomes. I didn't show you. We characterize it using micrococcal nucleus, so we can see 2, 3, 4 of the ladder. We're not doing salt dialysis. We're doing some using the ISY complex and NAP1. So we think this should help the nucleosomes get to close to what the physiological position might be. Remember, we do have 5 GAL4 binding sites, so it's not unlikely that at least some of them might be in linker regions as well. And maybe once you get one in, that helps to get the others in. It's a good question. I don't have a precise answer, but in this system, it's not the case that the activators are blocked from binding if the nucleosomes are there.
But there is endogenous ISY CHD activity in your extract.
Yes, yeah, but we can do the binding of the GAL4 VP16 is recombinant. So even if we do that without any activator, we still see it binding. Yeah. Yeah, question.
Do you see the first of all, really good talk.
Thank you.
I really love the single molecule microscopy.
Thanks.
Very fascinating. And I was just wondering about the single molecule microscopy. Do you see any parts of the transcriptional regulation that is being missed by this approach? I mean, you mentioned the RNA condensates, for example.
Yeah, well, so we haven't seen any evidence for any kind of condensates. And again, my take is in vivo, it's not clear to me how prevalent those things are. So the condensate literature, if you read a paper, there's usually 2 parts. There's something where they take a purified protein at high concentration and get some sort of droplets or gel in vitro. And then there might be some microscopy where they show you some clusters. But if you ask, well, what tells you those are the same thing, that's usually lacking, right? If there was a lot of clustering, I think we'd see multiple polymerases coming in all together rather than the sequential assembly. One caveat, though, of the in vitro system, even though this is nuclear extract and we think everything's there, and when we calculate the factor concentrations, we're probably about an order of magnitude less than the nucleus. I mean, nuclear concentrations are really high, especially in yeast nuclei, which are really small. So it is true that we are not quite at physiological concentrations. That might be one reason, if they exist, we might miss them. I don't want to rule that out. But again, I think the onus is on the condensate people to really prove that what they're seeing in vitro is the same as what the clusters are in vivo. But are there other things we're missing? Well, actually, what we haven't been able to see, and so far the yeast nuclear extracts don't seem to be so great at reproducing termination pathways. We haven't pushed very hard, but dabbling in a few experiments, it might be that the salt concentrations or cofactors, something might be slightly different. These extracts are optimized for initiation and elongation because we're just doing runoff transcription. It's certainly possible that if we played with conditions, if someone was ambitious, we could maybe get transcription coupled termination, but that's something that's been elusive so far. Yeah, Minkyu.
So, I mean, old models from jaundice and then Drosophila hisha polymerase tend to pose and then right after polymerase initiation, right? Just proposing that actually the duration of Pol2 dwelling time is not actually posed and actually terminating. Is it true?
Yeah. So John and Karen and other people have tried to do some measurements of what the half-life is of some of those polymerases, and there's one paper from Karen that's particularly nice, and it's a mixture, right? So there are, in that data, which is based on cross-linking and ProSeq and things like that, there do seem to be some promoters where the polymerase seems to be long-lived, and by long-lived, I mean maybe 10 or 20 minutes. The vast majority are on the order of minutes or so, which, as you know, is probably also the time scale for termination. So again, they could be paused, but many of those pauses might be pausing because that leads to termination rather than elongation. And then the other little bit of evidence that these things are turning over in vivo, Karen had a paper where they sequenced the short transcripts from the paused polymerases. And they did some calculations of how many copies of those short RNAs are there per cell. And so if they were coming just from the paused polymerases, you might say, okay, maybe there's 2 or some small number of these RNAs per cell. But in many cases, it's actually dozens or hundreds, which again tells you those polymerases are not taking all those short transcripts and elongating. They're turning over and spitting out these transcripts, and that's why they're in excess to the number of promoters. So. Okay, no other questions? Thanks very much. Thanks for coming out.